Hi! The previous videos have shown you the different cells in the nervous system and what a neuron looks like. In this video we'll look at how neurons talk to each other. We will also look at what makes the brain so special. How can a collection of cells do something special like making important decisions? My own research focuses on the neurobiology of addiction, so I use drugs of abuse as an example to show what happens when neural communication is changed. Drugs change how neurons communicate. It changes the way you feel, the way you behave, and how you perceive the world. So I'm going to use drugs of abuse as an example of what happens if neural communication is changed. Drugs change the way neurons communicate, the way you feel, how you perceive the world around you, and the way you behave. So this is a great tool to show you the complexity of the brain. Mario has shown you the anatomy of a neuron, including the axon and the dendrite. In this lesson, we will look at how a message is transmitted between neurons. So let's introduce the synapse. The synapse is the area where information from one neuron is transferred to another neuron. There are three important parts to the synapse. The presynaptic neuron is the neuron transferring the message. The postsynaptic neuron receives the information, and the little gap between them is called the synaptic cleft. The neurons are very close, but they do not touch. So there needs to be something to get the message across the synaptic cleft. This is where neurotransmitters come in. Neurotransmitters are chemical messages that are released by the presynaptic neuron when it's sufficiently stimulated. The neurotransmitter travels across the synaptic cleft. Neurotransmitters are like keys that fit to a specific lock. These locks are located on the postsynaptic membrane and they are called receptors. When a neurotransmitter finds a receptor that it can bind to, it will connect to it. And when enough neurotransmitters have bound to receptors, a signal is generated and will travel along the second neuron. The neurotransmitter is cleared out of the synaptic cleft and the process can start over. So let's look at an example. Dopamine is a neurotransmitter in the brain that's involved in activity, motivation and reward. When you do something you really enjoy, dopamine will be released in specific brain areas and give you a feeling of pleasure and will make you want to do it again and again. Let's look at the neuron that releases dopamine. There are a number of important steps. First, dopamine is built from other building blocks and transported to the postsynaptic neuron. Dopamine is stored in vesicles near the end of the neuron, ready for release. When an electric signal arrives, these vesicles with dopamine are released into the cleft in one big push. Dopamine moves across the cleft and binds to the dopamine receptor and activates it. When a receptor is activated, an electrical change takes place. If enough receptors are activated, the signal is strong enough and an action potential will travel down the postsynaptic neuron. The next step is to bring all the processes back into starting position by removing the dopamine from the cleft using breakdown enzymes and by reuptake of dopamine into the presynaptic neuron. For example, when kids swing on a swing or play video games, they are enjoying what they do, so their brain releases dopamine. And this reinforces their behavior, helping them to stay focused, want to do it again and again. Drugs like speed work in a similar way, but they have such a powerful effect that it brings your brain out of balance. When you take a drug like speed, your neurons will be stimulated so strongly that all the dopamine is released into the synaptic cleft. The cleft is now flooded with dopamine. All the receptors will be activated and it's difficult to clear the dopamine out of the cleft. Speed is clever as it affects the presynaptic neuron in several ways, making the dopamine linger even longer. The problem is that having such a strong stimulation changes the normal function of your brain. So how can an electrical signal change our behavior? This is where we need to look a bit better at the brain. The brain is not just a collection of neurons. We know 7 different areas in the brain that all have specialized role and functions. So when we notice a bee, one area in the brain registers the shape, one area the sound, and the other one how we feel anxious. So let's look at the different areas of the brain we know. On the outside of the brain we find the cortex. This is divided in a left and right side, the left and right hemisphere. There are 4 different lobes in this cortex. the frontal lobe, which is important in planning and analysis of consequences, the parietal lobe, important for math and spatial abilities, the occipital lobe for vision, and the temporal lobe for language and auditory information. The following system is the limbic system, which is important in emotion and memory. The next one is the cerebellum for motor control and motor learning. And the last one, the brainstem and the spinal cord. These are vital for brain communication with the body and the muscles. All these areas need to work together for the brain to function optimally and to respond quickly and accurately to any challenge. Our lives depend on keeping our neurons and brain healthy, and this is where support cells help us. We also have special protective packaging to make sure the brain does not get damaged when we bump our head. It floats in cerebrospinal fluid and is protected by membranes in our skull. This week's assignment will look at what happens when we bump our head or are in an accident. Associate Professor Corina van der Heuvel, an expert in this area, will tell you all about the assignment. You can also try the knowledge test to see if you're understanding the topic. Thank you for your time, and I hope you enjoyed learning about neural communication and the brain.